Somehow I forgot to cover this slide on the last lesson, so let me go ahead and get into this one before we get into the rest of the lesson here, which hoping with any luck we can finish this guy and get you guys ready for your test. So if you look at the two vultures up here, which I may have mentioned earlier, it's just all these days are kind of blending into one now. If we look at this guy and this guy, you would be very prone to classifying them as related. In fact, more so than you would classify either of them to being related to the stork. But we have to remember that this stork and this vulture have evolved in North America, whereas this vulture evolved in Africa. And in having done that, the two American birds came from a similar gene pool on the American continents, and the vulture came from a gene pool in the African continents a long time ago. And as they evolved, the environment predicted the outcome. Essentially, the vultures lived in environments where this phenotype was beneficial, and the stork lived in an environment where its phenotype was beneficial. And the other piece of this puzzle is that there are storks in Africa, or stork-like species, that would be more closely related to the African vulture. And DNA helps us clarify that. And that's why this slide was kind of crucial to our understanding there. Similar relationships exist down here in the pandas and some of the bear species. The raccoon is, by phenotype, more closely related to the red panda than the giant panda is to bears. And the problem with this is the naming of the panda bear and the red panda. The red panda is more of a raccoon. It shouldn't be called a panda at all. And that's one of the issues with the naming system and using common names. So I had to recap that for you. At this point, you probably would have finished this and hopefully had an opportunity to figure out that slide. So moving into the last section here, this is about the actual naming of these classifications, okay? So during Linnaeus's time, obviously, there were two groups of organisms, all plants and all animals, and that made sense. But what that leaves out are the fungi. And obviously, I think you guys might be thinking microscopic organisms, but this was before we were really looking at bacteria as a life form, because we really didn't have the microscopy techniques to get to that stage yet. So it's plants and animals. And it was logical to put the funguses, like mushrooms, into plants because, shucks, they didn't move, right? And animals did. But the major classification that we use now, fungi have their own kingdom, and it actually sits in between plants and animals because plants are photosynthetic, whereas animals consume food, and fungi actually consume energy from their environment as well. Wouldn't call it food, perhaps. It's not ingested like we ingest our food, but they don't use the sun. They consume calories, and in doing so makes them more like animals than plants, which I referred to on that last presentation right about here when we talked about this section. Why we are more close related to fungi, it's because they consume their calories. Okay, so looking at this, in the 1700s, Linnaeus's time, plants and animal kingdoms. And that's cool. In the late 1800s, we started to group all microscopic organisms into one group, plants into another, and animals into their own. Obviously, you can see that animals have a pretty consistent flow because they were more easily identified. By the 50s, the kingdom Monera came out, which included, at the time, we didn't know this, but eubacteria and archaebacteria, protists, which we're going to talk about in a minute, got their own kingdom. And then fungi, this was the first time we start to see fungi separated from plants because right in the 1950s, we started to study photosynthesis or began to understand it to a better degree. Okay, and now by the 90s, it was back when I was in high school, we had the six kingdom system. So this is nothing new, except the protists are still a mystery category, as you're going to see here. So major differences in the kingdoms. Let's look right here. Prokaryote. These are all eukaryotic, as you can see below. What separates these two is this molecule, peptidoglycan. So one group has it, one group does not. Cell structures, what you're going to find in eukaryotes, there's cellulose is going to be found in plants, and some protists. Animals don't have cell walls or chloroplasts. Plants do. Fungi cell walls are made of chitin, and we find that in some animals. Uh-oh, starting to get a little muddy here. Unicellular. Most of these are unicellular. Some are multicellular, which now we're getting related to fungi, but then, multicellular plants, multicellular protists, all right. Autotroph or heterotroph, same here. Plants, energy from the sun. Protists, both. Damn it. Heterotroph in animals and fungi. I mentioned that to you already. So why does this make things difficult? Let's follow this group of protists. These are plant-like protists. These are protists that exist kind of between fungi and plants. And these are animal-like protists. So what is the protist category to us now? It's a catch-all. It's where we put organisms that we're not really certain where they belong. And in the future, if we find some other means of classification, probably through DNA, then we'll be able to more accurately classify them and not have protists in 3 major branches, which is not what the idea of classification is at all. But many times, as we discovered here, it's a best fit scenario with given information. Okay. This is going to be a study using a dichotomous key, and it's a technique that we use to identify organisms. I don't know if y'all have ever experienced what a field guide is, but people who want to learn how to identify birds, they can travel around with a field guide. It's not just birds, plants, animals, anything. And it's specific to regions or certain locations within certain regions. And it starts with the likelihood of a particular organism happening, and then they put them all in this book, and then you can study them in this book. So you ask a series of general questions. So a general question that a dichotomous key could have, is it plant or animal? And that eliminates a group, and you start to get a little bit closer to what you're trying to classify with these questions, okay? So you're going to be able to do this at home just using this page. And if you want to, like I said over here, you can kind of click on this guy, and it's supposed to enlarge. Oh, it says enlarge. Good. All right, let's do this. Move this over, bring this guy out. And this is going to kind of, if you want to do this at home on your laptops, we're going to follow it from these perspectives. So compound leaf versus a simple leaf. If y'all want to use this one, you can to help you out. You're just going to ask some series of questions, okay? Is the leaf compound or simple? So if you work with me, let's find all the compound leaves first. A leaf divided into leaflets. So this guy's compound, this guy's compound, and this guy's compound. There's 3 of them. The other 4 are simple leaf, not divided into leaflets. So if you're going to pick an organism, let's just pick number 1 here, because y'all do that one first. So it's a simple leaf, and the directions tell us go to step 4. All right, so we go to step 4. The leaf veins, do they branch out from a central point, or they branch off a main vein? So if we see here, we see this main vein, and we've got branches coming off the edges of this. That's branching off a main vein. So if we come to this step, we go to step 6, a leaf edge. Is it jagged or is it smooth? And right here, this looks jagged. So number 1 is going to be betula or the birch leaf. Then you go on to number 2, 3, 4, and so on, and identify each one. And you can share those findings with me when you do your homework here. Okay? All right, from this point, you're going to work on finishing your study guide, getting ready for your test, emailing that group email with questions for everybody to see, and preparing to take another test at home. That's what we got to deal with, so you'll do your best. I know you will. I have confidence. And we'll check in as we go. Bye.